Hello viewers, this is Dr. Naina Khullar, Assistant Professor, Department of Zoology, Gujri College, Fatehgarh. I welcome you to the practical video lecture series on action of salivary amylase on starch, Part 1. The aim of this experiment would be to study the activity of salivary amylase on digestion of starch-rich foods, and the learning outcome would be that the students would be able to understand the process of digestion of starch. In short, how breakdown of polysaccharide, say starch, to a disaccharide maltose through salivary amylase or ptyalin enzyme actually happens. Before that, we need to know what are starch-rich foods. Starch-rich foods are basically plant-based carbohydrates like rice, potato, wheat, etc. Also, the foods that we binge on, the processed foods like noodles, pasta, macaroni are all rich in starch. Plants usually store glucose in the form of starch, while animals do that in the form of glycogen. Let us first understand the process of digestion of starch by salivary amylase. What is the role of saliva in digestion? How is starch digested in mouth? And how do we detect whether the starch is digested or not? Starch is basically composed of glucose monomers and they are linked by alpha-1,4 glycosidic bonds. These glycosidic linkages are actually broken up by the amylase enzyme which is present in saliva. This amylase enzyme breaks the bond and converts starch into a disaccharide named maltose. And that is the same reason after chewing the food for a while, we start feeling that the food is sweet. Humans have 3 salivary glands to produce the saliva: parotid, sublingual and submandibular. And all these produce saliva which contains the enzyme salivary amylase or ptyalin which helps in the digestion of starch. How can we test whether the given foodstuff has starch or not? This experiment can be performed at home if you have iodine solution with you. Simply take a slice of potato and add few drops of iodine solution to it. You would see soon it turns blue in color. This simply shows that your potato is rich in starch. If the same experiment we supposed to perform in the laboratory, how would we do it? So for that we need certain requirements. Test tubes, iodine solution, 15 milliliters of 1% starch solution, 3 milliliters of 0.9% sodium chloride solution, saliva and dropper. Procedure. We'll have to take 2 test tubes and mark them A and B. In both the test tubes A and B, we would be adding 3 milliliters of 1% starch solution and 2 to 3 drops of 0.9% sodium chloride solution. Now rinse your mouth to avoid any sort of food particles and spit out saliva in a test tube. 5 to 6 milliliters would be good enough. Now to the test tube A, add 1 milliliter of saliva solution. Keep it on the test tube stand for 4 to 5 minutes at room temperature. In test tube B, all the same stuff, that is starch, sodium chloride solution, is added along with the starch solution. Salivary solution is not added, which is the only thing which is omitted in test tube B. After 5 minutes, you have to add iodine solution to both tubes A and B. Let's see what observation we get. Test tube A shows no change despite the fact that we've added iodine solution to it, while test tube B gives blue coloration. The inference is salivary amylase has broken down starch into maltose in test tube A. Therefore, if we add saliva on starch, the salivary amylase present in saliva gradually acts on starch and converts it into maltose. Starch keeps on giving blue color with iodine till it is completely digested into maltose. Therefore, at the point when no blue color is formed, the starch is completely digested into maltose, which does not give blue color with starch. This is the end point or the achromic point. I hope you could understand the concept of salivary amylase and the breakdown of starch into maltose. Thank you.